Characters and classification of phylum Porifera. The porifera may be defined as asymmetrical or radially symmetrical multicellular organisms with cellular grade of organization without well-defined tissues and organs, exclusively aquatic, mostly marine, sedentary, solitary of colonial animals with body perforated by pores, canals and chambers through which water flows, with one or more internal cavities lined with choanocytes, and with characteristic skeleton made of calcareous spicules, siliceous spicules of horny fibers of sponging. General characters. Porifera are all aquatic, mostly marine except one family Spongillidae which lives in freshwater. They are sessile and sedentary and grow like plants. Body shape is vase or cylinder-like, asymmetrical or radially symmetrical. The body surface is perforated by numerous pores, the ostia through which the water enters the body, and one or more large openings, the oscula by which the water passes out. Multicellular body consisting of outer ectoderm and inner endoderm with an intermediate layer of mesenchyme, therefore diploblastic animal. The interior space of the body is either hollow or permeated by numerous canals lined with choanocytes. The interior space of sponge body is called spongocoel. Characteristic skeleton consisting of either fine flexible spongin fibers, siliceous spicules or calcareous spicules. Mouth absent, digestion intracellular. Excretory and respiratory organs absent. The nervous and sensory cells are probably not differentiated. The sponges are monoecious, reproduction both by asexual and sexual methods. Asexual reproduction occurs by buds and gemmules. The sponges possess high power of regeneration. Sexual reproduction occurs by ova and sperms. Fertilization is internal, but cross-fertilization occurs as a rule. Cleavage holoblastic, development indirect through a free-swimming ciliated larva called amphiblastula or parenchyma. 48. The organization of sponges has been grouped into three main types, viz. Askin type, cycon type and leuconoid type due to simplicity in some forms and complexity in others. Classification. The classification of porifera is based chiefly on types of skeleton found in them. Class 1, Calcalciaspongiae. L. Calx lime, or L. Calx lime plus spongose sponge. They are commonly called calcareous sponges. Skeleton consists of monaxin or triradiate and tetraxin spicules of calcium carbonate. They are dull-colored sponges less than 15 centimeters in size. They are solitary or colonial, body-shaped vase-like or cylindrical. They may show asconoid, cyconoid or leuconoid structure. They occur in shallow waters in all oceans. Asconoid sponges with radially symmetrical, cylindrical body. Spongocoel is lined by choanocytes. Cyconoid or leuconoid sponges having vase-shaped body. The body wall is thick and folded, choanocytes line only radial canals. Asexual reproduction by budding, the free-swimming larva is called amphiblastula. Example, Leucoxylenia, Clathrina, Cyton or Cypha, Grantia. Class 2, Hexatinelidaorhyalospongiae. G. Hyalus glassy plus spongose sponge. They are called glass sponges. Skeleton is of siliceous spicules which are triaxin with 6 rays. In some the spicules are fused to form a lattice-like skeleton. There is no epidermal epithelium. Choanocytes line finger-shaped chambers. They are cylindrical or funnel-shaped and are found in deep tropical seas. They grow up to 1 meter. Spicules are hexasters, i.e. star-like in shape. They are not attached by root tufts but commonly attaches to a hard surface. Radial canals or flagellated chambers are simple. Spicules are amphidiscs, no hexasters. They are attached to the substratum by root tufts. Example, Eupoctella, hyalinema. Demos spongiae. GR, Demos frame plus spongose sponge. Contains the largest number of sponge species. Large-sized, solitary or colonial. Asterisk the skeleton may be of sponging fibers or of sponging fibers with siliceous spicules, or there may be no skeleton. Spicules are never 6-rayed. They are monaxin or tetraxin or differentiated into large megasclerae and small microsclerae. Body shape is irregular and the canal system is of Leucon type. Generally marine, few freshwater forms. Sponges are mostly solid in simple rounded cushion-like, flattened in shape, usually without branches. Skeleton comprised mainly of tetraxin siliceous spicules but absent in order Myxospongida. Canal system is leuconoid type. Shallow water form. Simple structure. Skeleton absent. Spicules are not differentiated into megasclerae and microsclerae. Asters may be present. Spicules are differentiated into megasclerae and microsclerae. Example, Spongilla, Shalina, Spongia.